All right, so now we get in to the second video, which is transport across the membrane. Now this is a big one, and hopefully it looks familiar. If you don't remember everything, again, that's okay, but hopefully it doesn't seem completely foreign. So let's go ahead and talk about what we're going to get into here. We're going to talk about active versus passive transport. We're going to define some terms like solute, solvent, solution. We're going to talk about the idea, the concept of a concentration gradient, since that is extremely important in terms of how things move. We'll look into how the mechanisms of transport across a membrane, look at some examples, and then get into osmosis and how solute concentration or tonicity of the environment outside of the cell influences the cell, get into absorption and reabsorption, and then how do we get things across epithelial membranes through paracellular and transcellular mechanisms. So we are going to introduce some new terms terms maybe you've seen before. I'm not sure, at least in biology, I know I don't get too deep into this in earlier classes. So if you've seen these, you've probably only seen them in chemistry. But let's go ahead and get into them. So we need what's called a constant osmolality. Basically, it means the solute concentration, the things dissolved in the fluids, have to be maintained, right? We can't change solute concentration willy-nilly because that will damage cells. It can cause them to die. And so we are constantly trying to make sure that the solute concentration is kept relatively constant. And so this can we can achieve this through a couple things, right? We have osmoreceptors found in the hypothalamus that are going to detect a change in osmolality, basically osmolality going up. So this means solute concentration going up. That means you have more solutes compared to the water that you had previously, right? So your water's gone down, solute concentration's gone up. A lot of times this is due to dehydration. And so two things are going to happen in response to osmoreceptors detecting this. You're going to feel thirsty, right? And so you have that drive to get something to drink. And your water excretion in your urine is going to go down. So we're going to use a hormone called antidiuretic hormone, or ADH, to retain more water, pull it out. And so this is when you'll notice, like, if you've been dehydrated, one, you feel thirsty, and two, your urine is more concentrated, so it's very yellow. If you're really dehydrated, it can start approaching even like deep yellow, orangish, and if you're looking at extreme cases, I mean, you're looking at like deep colors, including like a brown, almost like a yellow brown. That's extreme, right? A lot of people today are probably dehydrated on a regular basis. But it's something that we can easily just combat, right? Just by simply drinking more water and listening to our body when our body says we're thirsty. So basically we're gonna increase the intake of water as well as prevent the loss of water at the same time if we notice that the solute concentration is higher than it should be. Once the plasma osmolality, that solute concentration, drops back down, osmoreceptors are no longer stimulated, and so we don't feel thirsty anymore, right? And then we can go back to normal of letting water enter into the urine. And so if you're really hydrated, especially if you're really, really hydrated, you'll notice that your urine gets more clear, right? And again, that's just because more water is being excreted out. So we have a way, right? Because we don't want to go too far in either direction, right? Maintain that balance, homeostasis, right? We can basically help control how much water we have and control this osmolality by controlling our thirst drive and controlling how much water ends up in the urine. Right, and so over on the right here we see the feedback loop, right, over here. And again, it's a negative feedback loop, right? How do we know that? If let's say our blood volume goes down, plasma osmolality goes up, so that we've lost water and we have more solute, or our solute concentration is higher, right? We drive the thirst mechanism, so we drink more water. We secrete ADH from the posterior pituitary to tell the kidneys to retain more water. We retain more water, we take in more water, and that's going to then, once we reach a normal level, right, for osmolality, then we turn that off, right? So our water was going down, We then enact activities, behaviors that will bring water up. And then once water level is high enough, we can stop that process. So again, it's a negative feedback loop that we see here. Now remember from the last I guess you say chapter, last section, right, a few videos ago, that our plasma membrane is selectively permeable, meaning there are things that can get through it. It's not an impenetrable wall, but there is nuance and there is selection in terms of what can actually get through easily. And that some things will be able to easily get through, other things will need a little bit of assistance, and other things just not at all. So this is to help limit the transport of substances in and out of the cell, and it gives us more avenues of regulation. Generally speaking, there are like two rules, right, to being able to pass through the phospholipids, right? And we see that in the picture here. We see that if you are large, like this big macromolecule, you cannot get through you cannot get through between the phospholipids. So if you're large, it's a no. If you have a charge, like these little ions and molecules, you cannot pass through the phospholipids. So those are like the rules that I set up, is that if you're large or have a charge, you're gonna need another way in. Right? Those are the rules there. If it's something small and without a charge, like carbon dioxide, it can simply pass between the phospholipids, like this little one in purple. I don't know what it's supposed to represent, but it can pass right through between those phospholipids, no problem. So what do we do then about things like ions? What do we do about things like water that's polar? What do we do about things like macromolecules? How do we get those through? That's where we're going to use things like channels, right? Or in the case of water, a specific channel called an aquaporin, right? Channel proteins, carrier proteins, something along those lines to be able to transport across. Now, we've been saying the word solute, but what the heck is a solute? Hopefully it at least sounds familiar. I know I've talked about it in past classes, but let's get into the nitty-gritty and remind ourselves if we forgot. So there's really three words we care about right now in this particular video: solute, solvent, and solution. The solute is the thing we want to dissolve. So let's make it really easy. Let's say we want to make some salt water, right? The solute would be the salt, right? The table salt that we're going to add. The solvent is the substance doing the dissolving. So in our example of making salt water or saline, right, the salt is the solute, the water is the solvent, right? So solvent will dissolve something. And then when we look at the two together, that's our solution. So let's kind of spell this out real quick and easy. Salt is our solute, water is our solvent. When we combine them together, we have a saline solution. Right, so hopefully that kind of solves that mystery, right? Or helps you kind of think back and remember that those definitions. Now, obviously, salt is not the only solute. We see a lot of examples. It can be anything that's going to dissolve in that water, dissolve in the substance. So hormones, ions, amino acids, proteins, gases, sugars, right? All sorts of stuff can be dissolved into our solvent, which for the most part is going to be water. And so if we look at some solutions that we find in the body, that can be things like the plasma in your blood, the lymphatic fluid, right, in your lymphatic system, the cerebrospinal fluid we find washing over the brain and spinal cord. So we have all these different substances that are dissolved in these solutions, and we want to be able to control where they go, right? Now, there's different ways we can transport things across a plasma membrane. We can either do it passively or actively. Passive transport does not require ATP. So it's free, basically. It's free for the cell to do. It travels along a gradient, meaning we're going from a high concentration to a low concentration until we reach equilibrium. So that's what that means. The way I like to explain passive transport in general, it's kind of like if you were to go floating down a river, right? You're in your little inner tube, you're floating down the river. You require no energy on your part to move with the flow of the water. So think of passive transport as going with the flow. You're going with the direction of the gradient from high to low, right? And so you don't need to use any energy. It does not take effort on your part. There are a few different versions of this. There's simple diffusion, facilitated diffusion, and osmosis, right? And we'll talk about each of those in just a little bit. But we can also go against the current, right? So we have active transport. This does use energy, so we do need ATP, and we're going against the gradient. So rather than go from a high to low, we're going from low to high. And so we're not going to achieve equilibrium here. In fact, we're trying to prevent equilibrium with active transport. Right? This would be like you swimming upstream. You were trying to swim further up the river, and so you have to actively put in a lot of effort to make it upstream against the current. There's a few different types of this as well. There's active transport, where we're looking at the transport of small molecules. Vesicular transport, which is movement of large molecules. And there's a handful of different versions of vesicular transport that we'll look at throughout this lecture. So again, what does it mean for along or against a concentration gradient? So this has to do with, are we going from low to high or high to low? Passive transport along the gradient is from high to low. This is because it's easy to go from high to low because we're basically going to just spread out until everything is equally dispersed, right? So we're moving towards equilibrium, which will naturally occur. Active transport, low to high concentration against the gradient. We are not looking for equilibrium here. We are trying to prevent equilibrium, right? We're trying to prevent that from happening. We're trying to keep one item on one side of the membrane and something else on the other side, right? We're actively trying to separate things out, and so that takes energy. Another way to think of it is a room getting messy versus the time it takes to clean it, right? How much effort does it take for your room to get messy or your home to get messy? Not very much, right? It's very easy to just leave things wherever, right? It's very easy for that to occur, but it takes energy to reorganize things. So you can think of passive transport like things are just kind of going with the flow, ending up where they end up, versus active transport. You are actively trying to organize things, right? And that takes more effort. So what are some types of gradients in physiology? We have a chemical concentration gradient. This is where we're looking at a specific chemical and the concentrations we see on each side of a membrane. So, for example, glucose. How much glucose do we find on the inside of the cell versus how much glucose do we find on the outside of the cell? So we're looking specifically at how much of a chemical do we find on each side of that membrane. Versus the electrochemical gradient, this is looking at the electrical charge across a membrane. And so we'll see this one when we look at, especially we look at like the neuron and the action potential of a neuron. So we can look at the electrical charge we find using ions that have like sodium, potassium, hydrogen, they all have a charge, right? We can look at how their placement affects the overall charge of the inside versus the outside of the cell. So like if we're looking at the neuron, for example, there's a lot of sodium on the outside, there's a little bit of potassium on the inside. So overall, the inside, because there's not a whole lot of positively charged ions, it has a negative charge, whereas the outside has a more positive charge in comparison because it has more positively charged ions, right? So more positive charge on the outside versus the inside. Even though we're looking at two different ions, again, they're impacting the same thing in terms of the overall charge of the inside or outside environment. So again, the selectively permeable membrane has a few rules. One, again, if you are small, nonpolar, and uncharged, you're permeable. You get through simple diffusion. You can just push your way through those phospholipids. Remember that. If you are large or you have a charge, you're going to need another way in, right? You cannot just push your way through the membrane. But if you are small and you don't have a charge, right, like oxygen or carbon dioxide or whatever, you can just slip on past those phospholipids, no problem. Let's say you're small, but you have a charge of some sort. Water, glucose, urea, right? Ions like sodium and whatnot. You're going to need a transmembrane protein to get through using facilitated diffusion. So we still don't necessarily need to use energy to let these things pass through, but we need to create a special doorway. And then rule number 3, if you are large, regardless of your charge or not, you're going to need to actually use vesicular transport because there is no channel protein big enough to accommodate. So we're going to need something, we're basically going to need to create a pocket of plasma membrane to bring something in or push something out. So let's go ahead and talk about passive transport using diffusion. Now diffusion is just, again, going from high to low concentration gradients. That's all it is, going from high to low, high to low, high to low. And this does not require energy, right? Again, this is free for our cells. We can do this simply, or we can use facilitated diffusion. So let's get into those. So simple diffusion, we don't need any sort of transport device. It's just going to happen. We don't even need a membrane necessarily. This is something that you can see happen just by putting a little bit of food dye in some water. So we see, like the example up here, we see a dropper of some sort of dye being dropped into water, and we see that over time that dye, that blue dye, is dispersing through all of the water. That's simple diffusion. It's just basically the random collisions and interactions of the molecule that's moving around. They're going to just bump into each other and push away from each other until they finally have dispersed all the way throughout. Again, this is free because we're going from high to low. So what are some examples? These small nonpolar molecules like oxygen and carbon dioxide. So when we breathe, right, when we do that exchange of oxygen and carbon dioxide, simple diffusion is all it takes, right? Really easy. We can also use this with hydrophobic substances like steroid hormones. Because they're hydrophobic, they can pass right through the membrane, no problem. So this simple diffusion is gonna occur until we achieve equilibrium. Basically, each side is equal in terms of concentration, and then there is really, there's not no movement whatsoever, but there's a net zero of movement. What do I mean by net zero? So let's look at the left first, and then we'll talk about the right. So on the left, we see a higher concentration separated from a lower concentration, and so we see more molecules from the higher side move over to the lower side. But when we achieve equal concentrations, it's not that everything stopped moving. Things are still moving. It's still dynamic, right? But for every molecule, for every solute that moves over to the right, another one is moving to the left. So there's movement. It's just they've been dispersed so much throughout the solution that we don't notice a difference in concentration in any particular location anymore, even though things are still moving. Now, what are things that can influence diffusion and how fast diffusion happens? Temperature. The higher the temperature, the faster diffusion happens, because the movement of molecules is happening at a higher rate, right, when there's a higher temperature. Larger surface area, we can diffuse faster. And so this is another reason why we have a limitation on cell size, or we have special structures like microvilli to increase surface area if we need to move things across that membrane faster. Concentration gradient, the higher the gradient, the faster it diffuses. And then the size of particles, the smaller they are, the faster they can diffuse. And then what are we diffusing in? If it's like a gas, right, that's going to be the fastest. If it's a liquid, we're going to be a little bit slower, and if it's a solid, it's going to be really slow, right? We can see this if we did a comparison. Let's say we looked at how fast methylene blue travels through water versus an agar gel plate. We would see that it would disperse through the water at a much faster rate compared to the gel. Now, we already mentioned earlier that there are a lot of things that need to get through from one side to another, but they just simply can't push past the phospholipids because of that hydrophobic center of the phospholipids. Anything with a charge is not going to be able to do this just on its own. It's going to need help, and that's where facilitated diffusion comes in. So using things like aquaporins for water, using channel proteins or carrier proteins for charged ions, or decently sized molecules like glucose, we can move them across using this special basically doorway we've created. We have two different classifications. We have channel mediated and carrier mediated. So let's get into carrier mediated first. Carrier mediated is using a transmembrane protein, so a protein that spans the entire membrane. And the substance or the molecule we're trying to bring is actually going to bind to the transmembrane protein, to the carrier. So something like glucose, for example, is going to physically bind to that protein. And then when it binds, it's going to cause the protein to change shape. So think of it like those, those doors that are like the spinning doors. You push in, and then you're kind of in that circular space, and if you keep pushing the door and kind of change the shape of the door, then it opens up to the other side, but only one side is open at a time, right? You can't just open and leave the door open. I guess you can kind of think of it like that, right? The glucose goes in, it causes a change or movement in the door, and then it opens up to the other side. So we see there's like our example here, various glucose transporters, or glutes, glutes, whatever you want to call them. But glucose transporters. So we have like GLUT1 in the central nervous system, GLUT2 in the beta cells for the pancreas and the hepatocytes, GLUT3 for neurons, GLUT4 for adipose tissue, skeletal muscle, right? And so these will all help transport glucose. They're just slightly different versions, slightly different transporters. Now, channel mediated, this is where we're basically creating like an opening, right? We're creating openings. So we're not worried about a conformational change in this case. We're not worried about changing shape. And there's two classes of these. This is great for ions and this is great for water. We have leakage channels. This is always basically partially open, like you've left the door cracked, right? And this is going to allow for ions to move along their concentration gradient. So things can kind of leak in. It's not a floodgate, right? We haven't totally opened it and allowing everything to come in, but it's always a little bit open, so things can kind of just sneak in, right, on a regular basis. Versus a gated channel, like a voltage gated channel or a ligand gated channel, these are only going to open if they get the right key. So that key can be a chemical known as a ligand, so like a chemical signal, or an electrical signal. So we have a change in charge, right? And so those can serve as a key, depending on what type of channel. They can serve as a key to open the channel. It also means we have a way to close the channel when we don't need to open anymore. So here's our leakage channel. Again, super simple. It'll just kind of spontaneously open and close. It's always kind of somewhat open so things can leak in. So for example, potassium leak channels in neurons, they basically help maintain that resting potential of a -70 so that we don't acquire too much of that potassium, or we allow a little bit in so we maintain -70. So if we're moving right away from that -70, we can control, kind of let a little bit of potassium move through those channels to maintain that. Versus ligand gated, right? This channel is normally kept closed, right? And then a key is going to come along, whether that is a ligand, right, that is going to bind to it, causing a change in shape to allow it to open, or it can be something like a voltage gated one where we have a change in the charge of the interior of the cell or the exterior of the cell, and that causes it to open. So a good example of a ligand-gated channel is like a neurotransmitter like acetylcholine binding with its acetylcholine receptor, and that allows sodium to enter into a neuron. So in this case, right, acetylcholine is our key, right? The acetylcholine receptor is the lock. When they bind together, we open up the gate, we allow the sodium to get through. Now the voltage-gated channel is a similar concept, except rather than use a chemical to control whether we open or close it, we're going to use the change in the electrical current or the voltage instead, right? And so we're looking at basically what does the voltage look like around the channel. If it goes up a bit, we can open up, right? So we have things like voltage-gated sodium channels that open during an action potential. And this will allow the sodium to pass through into the axon of the neuron. Now, osmosis is a specific case of facilitated diffusion, but this is where the solvent is moving, in this case water, rather than the solute. So water is going to move rather than the solute moving. Everything we've talked about up until now, we've talked about solutes moving, like salts and glucose and right, all sorts of stuff like that. But now we haven't really talked about the movement of water. So let's say you have a solute that cannot get through the membrane, right? No matter what, we have no channel, we have nothing that's going to allow for that solute to get through at that moment. What's going to move instead? Water. So water can move. So what we see here, right, is we have a high solute concentration here, we have a low solute concentration here. We still want to equalize the solute concentrations on either side, but since our solute is not going to move, water is going to move in response. So we're going to drain water out from this side over on the left and move it to the right, right? And that's what we see over here. We see that the water level here has gone down, the water level here has gone up, but the solute concentrations have been equalized. So water is attracted to the higher solute concentration. And so water is going to move in response to what we call tonicity of a solution. So we're looking at the fluid around the cell, and tonicity is going to describe the solute concentration we find in that solution. So we can have isotonic, hypotonic, and hypertonic solutions. Those are all just describing the solute concentration in comparison to the solute concentration inside the cell. So in an isotonic solution, right, the solute concentration on the inside and outside is about equal. And so water will move in and out, but it's a net zero. Again, for every water molecule that leaves the cell, another one comes in. In a hypotonic solution, we're saying the high solute concentration is inside the cell, so it's a low solute concentration outside. Water is going to be attracted to the high solute concentration, so it gets pulled into the cell. If it's a really hypotonic, we can pull so much water in the cell that the cell will actually burst. This is called lysis. In the case of red blood cells, we're looking at hemolysis. And we see that picture down here, right? Our little diagrams, right? Our normal red blood cell, it's happy in an isotonic solution. When it's in a hypotonic solution, we see water flooding in. They look really fat and round. We see that some of them could even burst open. And then if we're in a hypertonic solution, like a really salty solution, for example, the high solute concentration is outside of the cell. Water gets pulled towards the high solute concentration. So we're basically dehydrate the cell because water is getting pulled out, right? And so our cell starts to shrivel and shrink, which this process is called crenation. And so they start to look kind of like little raisins in a way. Now, osmotic pressure, this is basically the force surrounding the cell that is required to stop osmosis. So this can kind of describe the osmotic pull of a solution. So the higher solute concentration is exerting a higher osmotic pressure. So like pure water has a pressure of 0, but as we increase the solute concentration, we increase the osmotic pressure. So basically, if we increase the solute concentration in the surrounding fluid around a cell, think of that being kind of like squeezing the water out of the cell, like it's pushing in on the cell and squeezing that water out of our cell. When we have a pressure of 0, it expands out. right? When we have an isotonic solution, right, then the pressure being exerted from the inside of the cell out and the pressure being exerted downward onto the cell are equal, so the cell's nice and happy, right? When you have a high solute concentration, the pressure pushing down on the cell versus the pressure like being pushed out against the like outward, the pressure being pushed in is very high compared to the pressure pushing out. And then if you're in a hypotonic solution, right, there's no real pressure being, or very low pressure being pushed on the cell. So the pressure on the inside of the cell is pushing out really hard. So that's why we see that expansion. Right? And that's why we see the water being drawn in. Now, some transport, again, needs to use some energy, right? Everything we've talked about up until now has been free. But now we're going to get into using energy. But like I said, some of this needs to use energy, and that's where we get into active transport. Remember, we're actively trying to fight against equilibrium. We are going against the concentration gradient. So we need energy and we need a pump. There's two types of pumps. There's an antiport and a symport system, right? So that's what we're looking at here. It says co-transport, counter-transport. I'm more worried. I'm just going to use antiport and symport. Or antiporter and symporter when it comes to something like an exam. Both of them, though, are going to use energy to move substances against a concentration gradient. We're just going to be looking at what things are moving in what direction. So let's look at an antiport, right? An antiporter. So basically, we are moving one thing in one direction and something else opposite, right? So here, like the sodium-potassium pump, this is your best example of an antiporter, is the sodium-potassium pump. Oh my goodness, no, you don't got to get crazy with it, but you should know what a sodium-potassium pump is and be able to explain it as an example of an antiporter. So let's talk about it. So here, right, we have 3 sodium ions on the inside of the cell that are going to be pushed out. So they're going to bind, right, and again, we are going against the gradient. Right, so we're pushing sodium from a low to a high concentration, right? So we are going to push the sodium into our pump. We have ATP that's going to cause a change in the shape so that we can open up the gate on the other side to push that sodium out. And now potassium can be brought in, right? So potassium, now we've changed, potassium's brought in. We lose the phosphate, so we dephosphorylate. We change back to our original shape so that the potassium is brought inside. So sodium enters into the pump, ATP changes the pump shape, sodium is pushed out. We're still left in that same shape before, potassium comes in, we dephosphorylate, we change shape back to normal, potassium is dumped inside. So you see how we're moving two things in opposite directions, right? That's why it's an antiporter, right? Because we're moving two things opposite of each other. Versus a symporter, right? A symporter is basically kind of a paired system in that we are going to pump something out to create a false high gradient. We're going to like artificially increase the gradient of an ion, and then we're going to use that to help bring something else in. So basically to help drive other substances against their concentration gradient. So something like we see sodium being used to help carry glucose in, right? So we artificially create a high sodium, like with a sodium-potassium pump, create that high sodium gradient outside the cell. It wants to move back inside the cell from high to low. Glucose is high on the inside, low on the outside, but we want to bring more glucose in. We basically pair the glucose and the sodium together so that as sodium moves from high to low, glucose kind of comes along with it, going from low to high. And we can do that with a lot of different things. So we can do that with glucose, we can do that with amino acids, we can do that with bicarbonate. There's a lot of things we can use to basically create like a way to guide something in, right? And again, most of the time, when we're looking at symport pumps or secondary pumps, these are using the effects of a sodium potassium pump of the antiporter. So keep that in mind. I know you look at this picture and you're like, there's no ATP, but this does use energy. It just uses energy indirectly because we're using ATP to create that false sodium gradient, right? To artificially, I should say, not false, but artificially increase that sodium gradient. So we're artificially creating a high gradient of sodium so that sodium naturally wants to come back in. And then we use this to have sodium kind of guide other things in against their gradient. So I know it's kind of weird. It's a little bit, it might be hard to wrap your head around. If you need help with it, let me know. But yeah, right. So basically this Symport is going to also use this guy here, right? It's going to use the antiport as well. We can also do bulk transport. Let's say we're trying to move something that's really big, right? It can't necessarily get through easily. That's where we are basically going to use the plasma membrane as a pocket to move things in and out. Exocytosis, we're moving things out. Endocytosis, we're moving things in. Really similar in how they function, just kind of opposite of each other. But you notice like we're creating like a little pocket of the plasma membrane and then pinching off to create a vesicle. Whereas exocytosis, we have a vesicle, it's going to fuse with the membrane and then kind of push its way out, right? Kind of like if you were turning your pocket inside out. What do we use these for? Exocytosis is great for secreting things like hormones, mucus, neurotransmitters, anything that we may be releasing in bulk. And then endocytosis is good for things like bringing in something large like phagocytosis that's used by like white blood cells. Receptor-mediated endocytosis, where we have receptors embedded in the membrane and we want specific things to bind to those receptors and then bring them all in at once. Or pinocytosis, we want to bring a lot of things in that are dissolved in the fluid. So we're basically bringing the fluid and everything dissolved in it together. So I already mentioned phagocytosis. This is like cell eating or eating cell, right? Again, so your white blood cells are great at this, or some of your white blood cells, I should say, are great at this, like the neutrophils. They're going to basically engulf something, so like a bacterial cell, creating what we call pseudopods to kind of extend and like create little plasma membrane extensions that can wrap around something to form a vacuole or a vesicle, right? And then once it's brought inside of the cell, right, our vacuole, we can then bring some lysosomes to it. The lysosomes fuse and dump their digestive enzymes in there to break everything down. Pinocytosis, think like cell drinking. So think like Pinot Grigio maybe, or Pinot Noir, depending on if you're a red or a white fan. But cell drinking, right, if that helps you remember, then so be it. But we're going to bring in the fluid. So we're bringing in a bunch of fluid with everything dissolved in it. So we create like the little pocket, right? All this fluid comes in and then we can pinch it off and bring it in as a vesicle. And then receptor-mediated endocytosis, we want to bring something specific in. So we have receptors embedded in the membrane itself, right? And then as things bind to it, we'll then form that pocket, form a vesicle, and pinch off inside the cell. With exocytosis, again, it's basically an endocytosis in reverse. We have a vesicle, it's going to fuse with the membrane, and it's going to spill the contents out into the space outside the cell. So we use this when we do things like releasing histamine, when we come into contact with an allergen. Our neurotransmitters will be released in the same fashion when we look at the neurons, when we look at the presynaptic neurons, which we'll see again later. And then we have transcytosis. This is where we're going to actually go through the entire cell. So we're going to come in on one end, and we're going to leave out the other side. So we've transported ourselves all the way across. We're spanning the entire cell. So this is using just a combination of endo and exocytosis to go from one side to the other. So like the epithelial tissue in your intestines are fantastic with this because they're absorbing the nutrients from the food you eat and then pushing those nutrients into the bloodstream. So when we're bringing things in, we call that absorption. So think like absorbing in the small intestines to bring it into the cells. We also have reabsorption, so something that is potentially going to be wasted that we want to bring back in. So absorption would be like something that's never been brought into the blood, into the body. It hasn't been brought into the tissues so the first time we were doing so. Reabsorption is something that was going to be secreted out and that we brought back in to keep. And again, we're using things like transcellular transport that we just talked about, transcytosis. We can also use paracellular transport where we basically squish between the gaps between the cells. So we see that here, right? Transcellular, we're actually physically going through a cell from, into the cell and then leaving the cell, whereas paracellular is like we're using like the little alleyway between them. So you think of like transcellular, it'd be like you walking in the front door, walking through the house, and then leaving out the back door, whereas paracellular, you'd be like walking between the houses.